Okay, so it is 12:01.
Reserve that seat.
Hey everyone, welcome to Dreycey Defense. I'm Raisa, I'm chairing the committee. We have here in the room the rest of the committee. I'm going to introduce them. So we have Doyle, we have Ryan, Robin, and Chris. The way we're going to proceed is that we're going to have Dreycey present. And that's going to take about 40 to 45 minutes. He would like to postpone the questions towards the end. There is a last.
Just to make sure that some time to definitely ensure that everybody has time to leave after the initial presentation.
So after the presentation, we'll have about 15-20 minutes of questions from the audience. We are also on Zoom, and we are being recorded, including visual recordings, so just to let you know. The device we have here is 360 degrees. And after the questions from the audience, we'll take questions from the committee, and I'll have a closed session with Dreycey, and a deliberation session, and everything should be done within 2 hours. Yeah. So yeah, Dreycey.
Awesome. Well, good afternoon, everyone. Thank you for joining. My name is Dreycey Albin. I'm a PhD candidate working in the Living Matter Lab, and today I'll be defending my dissertation. First, I'd like to mention thank you for joining me on what I'm assuming is most likely your lunchtime. I truly appreciate it. The title of my defense is the Phage Toolbox, Automating Phage Discovery Through Novel Software, Devices, and High-Throughput Methodology. The cornerstone of my dissertation focuses in on bacteriophages. These are viruses that only infect bacteria, so they're relatively safe for humans. They consist of a capsid that holds the DNA or encapsulates the DNA and a sheet in which it pushes it through. They also contain spike, base plate, and tail fiber proteins for binding to a bacteria, a host bacteria cell. So the perfect name for bacteria, Bacteria X. As the host binds to glycoproteins, proteins on the cellular wall of the bacteria, it injects its genomic information. Thereafter, it creates proteins that are needed for creating progeny viruses, the child viruses from this initial infecting bacteriophage. That eventually ends in a lysis of the bacteria, killing the host cell. This is the lytic cycle because these progeny viruses can go back and infect a new host bacterium. Another potential life cycle for these bacteriophages is where they inject genomic information directly into that of the bacterial genome. And this is called the lysogenic cycle, or the phage here is said to be temperate. Overall, phages kill bacteria, but that's the way I like to look at it. And in particular, this is useful for phage therapy. Shown here is a picture of a 15-year-old girl that was fighting for her life. On the left-hand side here are visual sternal wounds from a mycobacterium abscessus infection. So sternal wounds, and on the right-hand side is the CT PET scan showing internal wounds as well. This was damaging both her kidneys and her liver. Her doctor tried antibiotics. Nothing worked. So her physician ended up turning to a researcher at the University of Pittsburgh, Dr. Graham Hatfield, and they deployed a phage therapy. After 6 weeks, we can visually see that the sternal wounds started to disappear and that the internal wounds started to dissipate. This literally saved her life. This is a crucial potential alternative to antibiotic therapy. Not only for bacteria not responsive to traditional antibiotics, but also because they're bacteria specific. So typically with these phage cocktails, these bacteriophages will bind to specific types of bacteria, whereas traditional antibiotics might destroy even good bacteria in your gut or things that you'd like to keep. Secondly, they're optimized through evolution. Now, that is not typically the best word to use when it comes to evolution, but the reason I use that here is because there's proteins on both sides, CRISPR-Cas9 or Cas9 proteins in the bacteria for killing and treating bacteriophages, and the phage have anti-CRISPR proteins for fighting bacteriophages. So there's proteins on both sides that allow for this ongoing war between bacteriophages and bacteria. In addition, there's autodosing. The more bacteria you have in an infection, the more bacteriophage will be produced at that site. There's also low toxicity, they're relatively cheap, and of course, they're a natural product. So they're important. And during this dissertation talk, all of the work that I've done over the past few years at Boulder has been focused on phage therapy, and more importantly, how to discover new phages or work with them in the laboratory setting. The way that I did this is combining computer science, biology, and electrical engineering to answer questions in phage discovery. So how do we actually find these bacteriophages? Well, it starts off in the environment. You can imagine we can take a soil sample and we can sequence all of the genomic information or DNA inside that soil sample to find new bacteriophages. The output of these sequencing machines is actually fragments of a genome called reads. They're not complete genomes that are output from this. And because of this, one of the methods that we commonly have to do is filter out bacteriophage-only reads. How do we filter out only the reads corresponding to bacteriophages? And a question that I had in this work is, can a space-efficient data structure be built and used to filter out sequencing reads stemming from bacteriophages? To go about this research, I present PhageFilter. It's a highly optimized space-efficient data structure and software that can be used on embedded devices to filter out sequences corresponding to bacteriophage genomes. This is currently in the second round of review in BMC bioinformatics. Next is phage manipulation. Once you get a bacteriophage, how do you actually work with it in a laboratory setting? Well, this is actually how I started the PhD, and I'll tell you one thing, it takes a lot of time, and it takes a lot of effort as well. So what we'd like to do, or the question that I had when working with these bacteriophages, is can a digital microfluidics device be used to miniaturize and automate bacteriophage research? To go about this, I created a digital microfluidics device extension called the PhageBox, and use this to automate bacteriophage research in a miniaturized platform. This has been currently accepted into transactions of biomedical engineering. So congratulations to the co-authors on that paper too for all the great work. Lastly, after we understand these phages, can they be used for phage therapy? And what is the most effective way? You can imagine there are certain proteins that are involved in this process. And there's also toxic proteins that you might not want in a phage therapy. So with that, I had a question. Can machine learning models be used to annotate open reading frames in bacteriophage genomes, and thereby allow us to look at the proteins inside these genomes to see the efficacy for phage therapy? In doing so, I present PhageScanner, the third tool that I'll talk about. This is currently in review in PLOS computational biology. Another tool that I won't discuss during the dissertation is DebruijnExtend. The reason for skipping past this tool is, for one, time, to make sure that there's time for the defense. In addition, I presented this during the comprehensive exam, so the committee has seen some of this work, but I'd be happy to talk about it in the questions. Together, I like to think of all the tools that I've worked on as the Phage Toolbox, essentially encompassing a suite of tools, much like Adobe Suite or Office 365, that has many different tools that you can use depending on the area or the thing that you're trying to solve. But with phage therapy or looking at bacteriophages, we really start with the laboratory. And that's why the first project that I'll talk about is the PhageBox. So backing up a little bit, when I first joined the lab, I worked with a lot of bacteriophages in the laboratory setting. So here's actually a plate of bacteriophage growing. The yellow lawn there is bacteria, and the transparent plaque are the viruses or the bacteriophage eating through the bacteria, shown as the transparent little circles there. To actually get to this point takes a lot of effort, a lot of time, and Mycobacterium smegmatis takes several days to grow. Not to mention the reagents are very expensive. To grow Mycobacterium smegmatis, you need this media called 7H9, takes a bunch of albumin, and I could see the money start disappearing as I looked at the spreadsheet. Also, many of these tasks can be automated. So that is the key question in mind here. Can not only these be automated, but can they be automated on a digital microfluidics device? In particular, before talking about what digital microfluidics is, because I'm sure everyone in here is now curious, what are you talking about? Digital microfluidics depends on a phenomenon known as EWA, electrowetting on a dielectric. The idea here, shown in this GIF from Duke University, is as a voltage potential is applied, the droplets observe a wetting, or a spreading out effect. In practice, for creating a digital microfluidics device, this phenomenon is used on an array of electrodes, as shown here. For each of those electrodes, we can activate them to move droplets around. This allows for the 4 fundamental operations of liquid handling: mixing, splitting, merging, and dispensing the droplets. So we're able to take traditional liquid handling protocols from a laboratory and put it on a miniaturized chip. There are devices that are commercially available that perform this. One is the DropBot, so it connects to your host computer, and you can utilize this to move liquids around. Now, one problem here and challenge is this device does not allow for heating, which would be necessary for doing polymerase chain reaction or for incubating bacteria, for example. The second thing is magnetism. You might want some onboard magnetism to allow for mobilizing biologics. And the reason for doing this could be to purify DNA. For example, you immobilize DNA and then you clean it with wash buffer. With that, I present the PhageBox. So I created the PhageBox to allow for both heating and onboard magnetism to allow for giving the DropBot and any other open source digital microfluidics device extended capabilities. This is what it looks like in the laboratory. Essentially in the back, I have the DropBot. In front of that is the PhageBox that I developed, hooked, or connected through an adapter piece. I also have a graphical user interface for controlling the device and allowing for ease of use if a biologist, for example, wanted to use this. Here's a blown-up picture of the PhageBox in all of its beauty. And on the computer here, this is the graphical user interface. In the top, this shows the set and measured temperatures on the chip. So we can look at what the temperatures are on the microfluidic chip as it's running. And in the bottom panel here is an easy-to-use PCR configuration setting for end users. So if they want to specify what temperatures for different states of PCR, they can do so easily using this graphical user interface. This is what it looks like in action. Oh, it seems like it only wants to play something. The music really makes it, so as you can see, I can move around droplets on this digital array of electrodes here. So that's it in practice. This is actually a video from the device that I created, the PhageBox, just to give some background into how it works and what it looks like in operation. I developed the device in 3 different layers: top, middle, and bottom. And the reason for this modularity is to allow for, if I want to print the bottom panel per se, I can only print that without having to print the other layers of the device. If the top and the middle look good, I can modularly change the bottom and etc. for the other layers. This is the AutoCAD view of that, and the entire device is 3D printed. The most important layer of this device is perhaps the middle. The middle contains all the components for heating and magnetism. In particular, it has a temperature sensor and a Peltier element, both at the top and the front and the back of the chip. In addition, it has an electromagnet in the middle for mobilizing any types of biologics. What this looks like stacked up is, again, we have the front and back temperature modules and the middle magnetic module, and the digital microfluidic chip sits on top of this. So in practice, it kind of looks like this, where I would move the droplets either to the front or the back of the chip to allow for heating. So again, this can be used to incubate bacteria or for PCR. If I want to immobilize anything like magnetic beads or DNA bound to magnetic beads, I can move the droplet here to the middle and turn on the electromagnet. As a proof of concept, not utilizing the components just yet, I wanted to answer a simple question. Can this be used on bacteriophages in general? What's a good proof of concept there? Here, what I wanted to do was cut a bacteriophage genome, and the way that this is can be done in the lab is by using a restriction enzyme. This is an enzyme that will bind to some type of palindromic motif in the genome, causing a double-stranded break, as you can see here. In particular, I use the restriction enzyme BSU15I, and I mix this with genomic T7 DNA. After dispensing onto the device, merging the droplets and mixing them, by this point, the enzyme should have done its work in cutting the genome. I extract it from the device, and I test the outcome of that. So we're expecting this to cut the genome in 3 different places. I purposefully picked BSU59 for that reason. So 1, 2, 3, 4, 3 different places, and the result of that would be 4 different fragments from that. As I look at as I observe the electrophoresis gel here, I used a 1 kilobase ladder for comparison, 200 nanograms of uncut T7 genomic DNA. And in the last line here is the restriction enzyme the genome with BSU15I. And I indeed observed that there were 4 different bands. This is as expected because there's 3 different cutting locations with the restriction enzyme. So this indeed validated the expectation that the restriction enzyme digest works, and it's a good proof of principle that we can work with bacteriophage. We can do bacteriophage protocols on this device. So going into the components, again, I mentioned that there was that electromagnet in the middle of the device. I wanted to test that. The way that I tested this was using a magnetometer. I put it over the middle of the device, and I tested the magnetic strength under different conditions. On the left-hand side is the magnet inactive. So you can see that there's no magnetic field strength produced, if that makes sense. And on the right-hand side is with the magnet active, you can see an increase in magnetic field strength. So here, one thing to note is with the chip on there, there's actually a slight dampening of the field strength. This is kind of expected because there's something blocking the magnetic field strength. And I also used a stir rod for comparison. So a traditional magnetic stir rod. These aren't very strong. So the biggest question that I had here is, will this be strong enough to immobilize or localize magnetic beads on the device? To answer this, I simply mixed a droplet with magnetic beads from a Thermo Fisher kit, a biologics company. So I mixed those magnetic beads inside the droplet and I turned on the magnet. What I realized is the beads started to localize in the droplet. Now, if I move the droplet, the beads might go with it if the droplet's big enough. So how can I actually use this? Well, since they're immobilized, I realized the potential usage of this in addition to also using antibodies or any other type of immobilized biologic could be concentrating DNA. So if I mix DNA with these beads and then I split the droplet, that droplet containing the beads should have a higher concentration of DNA. A super simple proof of principle. That's exactly what I tested next. With the magnet off, the DNAs were evenly dispersed throughout the droplet. As I turn the magnet on, they localized near the magnet, and then I split the droplet. So then I take both the droplet containing the beads and not containing the beads, and I looked at them, right? I looked at the nucleic acid concentration using a spectrophotometer. So here I used a nanodrop 2000, which is the spectrophotometer, and I looked at the amount of DNA from all the droplets. Here, the concentrated DNA from bead split corresponds to the droplet containing the beads or containing the genomic DNA, and it had a higher concentration than the initial droplet. So indeed, we're able to concentrate genomic DNA on the device. The other way that this could be used, you can imagine, since we split the droplets, we can then add, if these are bound to antibodies, we can add some type of antigen to that droplet, then split the droplet and use that for observing any type of antigen inside of the device. The second thing I needed to look at was the temperature sensor. A common problem on digital microfluidics devices and microfluidic devices in general is evaporation. As you start to heat these droplets, they evaporate quite quickly. And the hypothesis that I had from reading literature is that glycerol might be able to prevent this, especially on this device. So looking at 0%, 5%, and 10% glycerol at different temperatures, 60, 70, and 80, I indeed observed that the evaporation rate was decreased as the temperature is heated. So here, using higher amounts of glycerol, let's say 10% for a polymerase chain reaction on the device, would help prevent this evaporation. One big question is, there's a temperature sensor and a Peltier element. So how do I set the temperature? Well, what I devised here is a strategy very similar to your household thermometer. If it's too cold inside, turn the heater on. If it's too hot, turn the heater off. This results in a bang-bang, a simple bang-bang controller where the Peltier acts as the heater and the sensor is the sensor. So if it's too hot on the chip, turn it off. If it's too cold, turn it on. And this is facilitated through adjusting a relay to turn on or off voltage to the Peltier element. To actually have this work with PCR, polymerase chain reaction, I embodied the polymerase chain reaction into a finite state machine. I used a finite state machine to embody the PCR reaction. PCR stands for polymerase chain reaction, and it's used to amplify DNA. So we'd like to do this on the device. Using this finite state machine, this was put onto the embedded device within the PhageBox, the Arduino Nano. And what it allowed for doing is controlling the temperature, cycle temperature states of each of these Peltier elements. Starting off with denaturation, this actually splits the genomic DNA or any other DNA that you're trying to amplify at 90 degrees Celsius. And then for annealing, this binds the primers to the DNA, and extension at 72 degrees Celsius allows for essentially amplifying the DNA using an enzyme called Taq polymerase. And it cycles through this, splitting and amplifying the DNA. Many times, you can say 32 times, and that's what we'd like to do on this device, this cycle temperature scheme needed for PCR. Using the graphical user interface I developed, I observed that the finite state machine indeed does work. This isn't the only time that I looked at this. There were multiple times that I looked at this, but this is just an illustration of that working. Here, the black line indicates, again, the set temperature. The green line is the measured temperature on the front of the device. We can see that it follows the temperature as required or set in the graphical user interface for polymerase chain reaction. To validate that the temperatures on top of the chip were correct, I used a thermography device to look at the extension, denaturation, and annealing temperatures, so 72, 91, and 46 degrees Celsius, and validated that the temperatures on the device were as desired. Secondly, lastly, I should say, I introduced a PCR framework. And the idea for this is to allow for PCR to eventually happen on the device with utilizing both the temperature modules on the front and the back. Here, because there's evaporation, I wanted to ensure that the PCR buffer on the front, right, kept at 50 degrees Celsius, could be used to replenish any media loss during the PCR on the front of the device. So this is kept at a constant 50 degrees Celsius, and as PCR starts to evaporate anything, then we can replenish that media using the PCR buffer heated in the back of the device. What this looks like in practice, here I put it through a pilot simulation using PCR buffer. The back Peltier element is kept at the constant 50 degrees Celsius, and when the front starts to evaporate, right, because that's performing PCR, I can replenish that droplet just in time during the annealing step, that's at 50 C, as it starts to evaporate. So it shows a framework for being able to perform PCR. Overall, in this work, I present a digital microfluidics extension that can be used with open source digital microfluidics devices. The novelty in this is the magnetic and temperature modules for being able to heat DNA or amplify DNA through polymerase chain reaction or mobilize biologics. The third thing that I present is a graphical user interface for controlling the device. So here, this graphical user interface allows for controlling PCR as needed. And overall, I use all three of these different deliverables here to show proof of principle concepts in bacteriophage applications. The second project that I'll talk about is PhageFilter. PhageFilter, or typically, if you want to discover new bacteriophages in the environment, you'll go and sequence a soil sample, or you can even sequence your gut microbiome. You can sequence different environments to look for DNA corresponding to bacteriophages. And a problem commonly addressed inside, and when working with this type of metagenomic data consisting of many genomes, is trying to filter out only bacteriophage reads or DNA. So here, that is the major goal of this project. How do we filter out these reads? But in particular, how do we do this on a small embedded device? So the idea for the software is not only to be able to do this using some commonly used bioinformatics tool, but to be able to make sure it's amenable to a small device, an embedded device, like a Raspberry Pi. The current methods for doing this are time-consuming, memory-intensive, and they're not going to scale massive metagenomic data sets. So the question I had before starting this project was, can a space-efficient data structure be built and used to filter out sequencing reads stemming from bacteriophages? There have been previous works that looked into doing this, in particular for filtering out DNA reads. In 2010, there was a paper published that used a Bloom filter. A Bloom filter is a space-efficient data structure that allows for answering a simple question: is an item in this set, or has this item been seen before? And that's how they utilize it for this application. In particular, Bloom filters consist of many hash functions and a bit vector in which they can store information into. So the idea of this in practice here is that, and I mentioned a hash function is a function that can map any type of input into a discrete set of numbers or a discrete output. And the way these hash functions are used here, we have two hash functions. We input a DNA sequence, GGAGATC. In the first hash function, we get 1. In the second, we get 7. Those hash values, the output of hash functions, can then be used as indices in the bit vector to add a 1. So wherever the hash function is mapped to, we add a 1 in the bit vector. That is how an item is added to a Bloom filter. Now, if we want to check if we've seen a sequence of DNA before, well, then we can actually use the same hash functions on an input sequence. So here, have we seen the sequence GCC before? After setting those to the hash function, we check the indices corresponding to those hash values, and we see that one of the indices is not set to 1. So it's 0 here, which indicates no, it has not been seen before. If we want to check the original sequence used for this example, the original sequence added to the Bloom filter, well, we can use the hash functions for that sequence, map to the bit vector, and we see a D or 1 set there. So it has been seen before. Now, there is a potential false positive rate with this, but not discussing that now, just looking at this, it seems that we have seen this sequence added to a Bloom filter. A novel piece of work came a little after this and isn't in the same area. It's focused on transcriptomics. What they did, though, Brad Solomon and Carl Kingsford, here they created a binary tree of these Bloom filters. And what was very cool about this work is now they're able to look at whether or not a transcript comes from a specific RNA-seq data set very efficiently. So it's a binary search tree that utilizes these Bloom filters. And the reason why I show this is I combined the ideas for filtering reads here using the original Bloom filter with the binary search tree of Bloom filters. In particular, again, I want to take out any reads that correspond to bacteriophage genomes. So I might have a bunch of bacteriophage genomes, and then I create the Bloom filters corresponding to those genomes. So here, the Bloom filter for this particular genome, it's a fingerprint for it, for the second genome. And now I can use those Bloom filters to answer the original question that I proposed. Given a read, can I check whether or not that read comes from any of these bacteriophage genomes? And if we recall the way the Bloom filters work, first I have to send it through the corresponding hash functions for those Bloom filters, and then I can check. At indices 2 and 5, are those bits set to 1? Well, indeed they are. We can see at indices 2 and 5, those are set to 1. So this read does potentially come from the bacteriophage genome. If I check the other bacteriophage genome, we see that indice 5 does not have a 1 set, so there's no way that it comes from that genome. The problem with checking these Bloom filters like this in this linear fashion is eventually it's going to be time consuming and expensive. So if I have a bunch of bacteriophage genomes and I need to check if reads correspond to any of those genomes or come from potentially any of those genomes, this is going to become a linear search. Utilizing the sequence Bloom tree, the way that this works is essentially by having internal nodes. So all the leaf nodes consist of genomes or Bloom filters for those genomes, and at the internal nodes are unions of any child Bloom filters. So these contain the union of all Bloom filters below it. Building out the tree for this entire thing would look something like this. At the root node here would consist of a union of all the Bloom filters in the tree. And again, that same property holds for any internal node below it. They consist of the union of all Bloom filters below that point. Now this works a lot faster because, again, I can take that same read and check if it's in any of those Bloom filters. I'll check the first one, the root node. If it's not in there, that saves us a lot of time because there's no way it could be in any of the genomes within the tree. Now that we see that it is in the tree, we continue going forward, right? Because 2 and 5 have bits set to 1. I check the child nodes of the root. It's not in the right child here, but it is in the left child. So then I check the child, the grandchild's, I guess, of the root node. And you can see on the right-hand side, the corresponding bits are set to 1 there. So again, this just allows for a faster way to check the genome. And the big help with this illustration here is the fact that we don't have to check the left side of this tree at all anymore because it's not in there. So this saves us time if needing to check a bunch of genomes, whether or not a read comes from any of them. This allows for a logarithmic time complexity as semi-blocked by the screen here, so I apologize for that. Taking a back step, what I present here is, or what I call this data structure is the genomic Bloom tree. So again, the original authors use this for transcriptomics. Here, I'm adding genomes to this data structure. And the goal is to take metagenomic reads and filter out reads corresponding to bacteriophages. The input here being metagenomic reads, the output being the filtered reads corresponding to bacteriophage genomes. So this acts as a filter. This genomic bloom tree acts as a filter to be able to filter out these reads. And in addition, I added the ability to look at what type of bacteriophages are in the sample and how much of those bacteriophages, taxonomic classification and relative abundance estimation. The novelty in this approach is one, it's applying sequence bloom trees for use in genomics rather than transcriptomics. It involves an improved filtering approach compared to the linear search method, and it contains a low memory footprint. And I'll get into this in a second, but again, I wanted to run this on small embedded devices, and the reason for this is portable sequencing. If you connect the Raspberry Pi, for example, up to some type of portable sequencing machine, then perhaps we could use that directly to filter out reads corresponding to bacteriophages. Just looking at the performance of the tool here with read filtering, again, the idea is to just filter out bacteriophage reads. As I increase the error, well, I compared to BioBloom tools and Phage. So BioBloom tools is a tool that came out much later. Phage is that original 2010 paper that I talked about. BioBloom tools is a newer paper. And just comparing the F1 score, essentially how well do these tools work at being able to filter out bacteriophage reads. As I increase the error in the sequencing reads, these are simulated sequencing reads. As I increase that error, the tools perform. They have a harder time of being able to pull out those reads, right? Because inside those bloom filters, they may have never seen any reads that look quite like that in a very high-level way to put it. But PhageFilter still performs just as well or better than the other tools. In addition, I tested increasing the contamination percentage here. A question I had is, well, what if there's only a couple bacteriophage reads in a giant file filled with a bunch of bacteria? Well, this is like pulling a needle out of the haystack. And the further the contamination percentage, the harder this becomes. And all the tools struggle at some point, like if there's 99.9% bacteria reads inside the sample, the tools struggle at only pulling out bacteriophage reads. So there's going to be some false positives in there. But PhageFilter does an incredible job even at this higher contamination percentage. So I also looked at classification, again here, answering the question, what types of bacteriophages and how much of those bacteriophages are in the sample? I compared against state-of-the-art tools, Fast Virome Explorer and Kraken 2, and again, I compared it with BioBloom tools. So BioBloom tools is another bloom filter-based tool. The tool does not perform anywhere near as good as Kraken 2 or Fast Virome Explorer. I do want to mention the F1 score here starts at 50, so that might make the axis look a little bit different there. But it performs a lot better than other bloom filter-based tools that are used for this approach. So here, BioBloom tools, one of the goals here is to perform classification. We still outperform that tool. Okay, one of the things I mentioned was running this on a Raspberry Pi or some type of embedded device, again, for potentially use of portable sequencing. And I realized some potential problems before actually applying the data structure on the device. One is that, well, it takes a lot of memory. So as I increase the number of genomes shown here in the rows, or the number of reads that I want to filter in the columns, memory is increased. So increase the number of reads or increase the number of genomes inside the data structure increases the amount of memory consumption. For that, to reduce the amount of memory consumption for the number of reads applied, I created a read buffer. This is a pretty simple approach where I pull in the reads, I read any type of genomic reads into memory in batches. So I pull in the reads in batches, and then I send them through the data structure to filter the reads. And this works very well in practice. You can see here in the rows, only the genomes increase the memory consumption, whereas the columns don't make a difference. So here, as I increase the number of reads or if the data file becomes bigger, it doesn't change the amount of memory consumption. The second thing is the number of genomes. As I increase the number of genomes in the data structure, this results in more bloom filters, right? So I wanted to only have a certain number of bloom filters in memory at once. The rest I save to disk. The benefit of this is by using, or what I use here is a least recently used cache, an LRU cache. So I only keep bloom filters that I've seen recently in memory. And there's a huge memory advantage here because I can choose the size of the cache. So only a certain number of genomes are kept in memory at once. And now we see that if I don't use the read buffer, only the number of reads play a role. So only the columns change, whereas the rows stay the same. If I increase the number of genomes in the data structure, no change. So this allows for extreme memory efficiency on the device. Using both the LU cache and a rebuffer, I can use extremely low amounts of memory while running the tool. And the reason this is important is because there's different types of compute that utilize more memory. A lot of bioinformatic tools might be developed for use on a laptop, and some even for usage on a supercomputer. For example, Kraken typically needs around 500 gigabytes of memory to load its full database, so it takes a lot of memory to be able to run some of these tools. Testing the memory performance of these optimizations, first I looked at how memory performs when building the genomic sequence bloom tree here. As I decrease the LRU cache size, the memory consumption starts to stay more constant. So here, decreasing the cache size from top to down, memory consumption goes down regardless of the number of genomes. I also wanted to compare against state-of-the-art tools that I used in the benchmarking, so Kraken 2, FACS, Cloud Tools, and Fast Biome Explorer. As I increase the number of genomes here during the query process, so all I'm doing here is querying the data structures that they produce, the memory consumption goes up for both Fast Biome Explorer and BioBloom tools. For the other tools, if I looked in closer, so they use a lot less memory than the tools just mentioned. So here, Kraken 2 and FACS also exhibit a linear increase in memory as the number of genomes is increased, while only PhageFilter is able to keep a constant memory size. So as the number of genomes gets to the thousands, this is going to be a problem for running it on embedded devices. As a proof of concept here, I actually connected a 4 terabyte disk drive up to a Raspberry Pi 3. So the Raspberry Pi 3 here has 2 gigabytes of memory. And then I use that to filter out bacteriophage reads from soil samples that I got that were originally extracted, not by me, by another author, from Dijon and Montpellier, France. But if anybody wants to pay for that trip for me to go there, I'll take it. So the accuracy here isn't necessarily an accuracy. I'd say this is an agreement with Megablast. So I compared the genus and species level assignments with Megablast. We can see that there is a slight difference in the species level assignments, but the genus level assignments were completely the same. So this is just a way for me to try and mentally validate that the output of the taxonomic classification is semi-accurate. Looking at the actual genus level assignments from the tool, for both the samples, they showed high levels of nonic virus and Surratt virus inside the samples, differing just slightly. In addition, the species level composition slightly changed. Not going into the species here, but the basic idea is the compositions are very similar. In addition, I was able to recapitulate the findings that they had, which were only at the family level. So I was able to have a finer grained taxonomic assignment here with the tool, going all the way down to the genus and species level, but also revalidating what they found in the original study. So here, PhageFilter allows for read filtering, classification, and low memory optimization. All right, getting into the last of my dissertation. Thank you for taking this roller coaster with me. Going back to the 15-year-old girl. Again, this is her with her mother on the right-hand side, Dr. Hatfold. This is the professor from the university and her physician. Her mother noted within weeks, this amazing treatment from Mother Nature was having this incredible effect on her body. But also noted in this article is we have not yet cured her. So there's still work to be done. In particular, from my viewpoint at least, it seems that the best way to try and improve the efficacy of these phage therapies and to learn more about bacteriophages in general is to go back into the way that they work in killing bacteria, looking at the proteins that are used for this process of eventually infecting and lysing a bacterial host cell. The proteins that are most responsible for this are called phage virion proteins. They consist of the capsid head, the whiskers, of course, the base plate, tail fiber, and spike proteins that bind to the bacteria. All these proteins are necessary to learn and find new ones because they correspond to how the bacteriophage infects a host bacteria. There have been tools, one thing that I want to mention here too is that there are many, I think 50 to 90% of bacteriophage proteins have not been characterized. So there's a huge need to be able to characterize these bacteriophage proteins. We're not going to understand what we're dealing with when using it in phage therapy. There have been tools to help solve this problem. So here are these tools. They essentially would take proteins from UniProt and they train binary classification models to essentially ask a simple question. Is a given protein a phage virion protein? So given a protein from UniProt it trains a model to answer this question. Is this protein a phage virion protein? So a bunch of tools, not all of them are shown here, go for the approach of binary classification. So machine learning approach to essentially say 0 or 1, is this the outcome? So is it a phage virion protein? A second set of tools recently developed, so a lot of these were released in 2022 and 2020, look at using a multi-class classification approach for trying to annotate proteins from bacteriophage genomes. In particular, if you get a bunch of proteins from, they use entrails as their database, they get a bunch of proteins from entrails, and they'll create models that answer the question, what type of phage virion protein is the protein? So here it's multi-class, but instead of just saying, is it or is it not a phage virion protein, we can say what type. Is it a capsid? Is it a teofiber, base plate, or spike protein? So they created this multi-class classification approach. There are many limitations of this work, though. The first limitation I realized was they only work with sequencing, as they only work with protein data as input. So they never use any type of genomic information. So there's no application to genomic data whatsoever in any of these prior work, which is, I feel a missing area because you might want to actually scan a genome to be able to learn more about proteins that haven't been characterized inside that bacteriophage genome. In addition, there isn't a framework for quickly developing these models. In addition to looking at phage virion proteins, what if we want to find phage proteins that are toxic? Or should it be used in phage therapy? It makes sense that these would be useful to find. The overall question that I had when pursuing this research is, can machine learning models be used to annotate open reading frames in bacteriophage genomes, much like the prior work using phage virion prediction? Here, I can predict the open reading frames and tools. Originally, I had an idea of scanning along these genomes, but that is quite an endeavor. So instead, using state-of-the-art tools for finding these open reading frames that eventually correspond to proteins, right? So these open reading frames turn into the phage proteins that I'm talking about. So what I can do is I can translate any open reading frames found in a genome into the protein and then use these models to be able to annotate these open reading frames. So one, I can ask the question, is it a phage virion protein for that open reading frame? What type of phage virion protein is it for that open reading frame? And I also introduce a BLAST classifier that allows for secondary validation. A lot of times machine learning models, they're not very transparent. It's not clear on why they show certain things. So being able to add another model, such as BLAST to this, allows for some secondary validation. To go about this, I developed PhageScanner in 3 different pipelines. The first pipeline is the data pipeline. The idea here is for both UniProt and Entrails, you can specify queries to pull proteins from those databases, cluster the proteins. I'll get into the details of this, but clustering the proteins is based on sequence similarity. So we'll go into different batches. And then I partition the clusters. The reason for partitioning the clusters is to ensure that between the training and testing data sets, there's a difference in sequence similarity between the proteins. So I don't want similar proteins in both the testing and the training for these machine learning models. The second pipeline is the training pipeline. So here, this is where I create machine learning models, excuse me, or an end user can use this to create machine learning models. So cleaning the proteins, so getting rid of any non-canonical amino acids from the proteins, extracting features. So this takes a protein sequence, and turns it into a feature vector that machine learning models can use, and train and test. So the train and test pipeline, again, trains these machine learning models and then test them to see how well they perform. This is very similar to prior work, except now it's embedded in a framework. The novelty in this work comes with using the prediction pipeline. So after we've tested and trained new models, here I use genomic information as input. Again, I want to stress that point. The difference here is genomic information. So we can put in metagenomic reads. I assemble them using a tool called MegaHit, which works very well for assembling metagenomic reads, and that's the reason I chose it. Secondly, we're finding open reading frames. This can be quite an endeavor. So here I use a tool, Annotate, that is specifically designed to find these open reading frames in bacteriophage genomes. I then translate those those open reading frames into proteins, and I use the trained models to assign features to each of the open reading frames. Is it a phage virion protein? Is it toxic? Should it not be used in phage therapy? The way that this works, first going into the data pipeline, is I use both Entrails and UniProt. Prior work has not used both these databases within this area, so a little bit of novelty there. And the way that the data pipeline works is for each of the machine learning classes, you can imagine major capsid, minor capsid, base plate for the multi-class model consists of queries to these databases. So I have for each of these labels or classes, I have a query that goes back to Entrust or Unipro. Again, to make sure that there's a difference between the testing and training data, I clustered the proteins using CD-HIT. So based on sequence similarity, they go into different clusters. Looking at the cluster size and the count of proteins per cluster, I observed that most of the clusters are below 400 proteins in size. So these clusters are relatively small, and most of them are very small, containing only a few proteins. Lastly, I adjusted the clustering identity threshold just to see how similar or different these proteins are. So at a clustering identity threshold of 90, there wasn't much of a difference in the number of clusters produced after that point. So the cluster count here essentially stops or doesn't really decrease much more than that after a clustering size of 90. So I used that while operating PhageScanner. I mentioned that I need to turn these proteins into feature vectors for usage in machine learning models. You can't take a string or a sequence and just put that into a machine learning model. You need to be able to allow it to be used by a computer. And the way that this is typically done is by taking sequence relevant information and vectorizing that, as shown here, a very basic, simple illustration. There are many ways to do that. One is tripeptide composition. So this takes a sliding window of 3 across a protein. And for any of the tripeptides in there, there's a corresponding index. So you can add the frequency to that index. Here, that vector would be 8,000 elements long. And the reason for that is there's 20 amino acids. So 20 times 20 times 20, 8,000. So that's the number of potential or different possible tripeptides. The model allows for easily testing a bunch of different feature combinations. And in addition to the model, Secure used a logistic regression model and tested the F1 score of these different feature combinations. So here, just testing all the different feature combinations, you can see that the TPC and DPC dipeptide composition both work very well for being able to be used as feature vectors. I didn't explain many of the different features. I'd be happy to talk about these in the questions, but the overall idea here is that these are used for turning a protein sequence into a vector that can be used by machine learning models. Okay, so utilizing the original pipeline that I showed, the training pipeline, I wanted to create different models for different classification tasks. First, is a given protein a phage virion protein? For that, I re-implemented several models from literature. So all of these models were re-implemented in the pipeline or in PhageScanner. This is one of the utilities of the tool, being able to easily re-implement machine learning models within a config file. So one, I also created a BLAST classifier. So this performs the best. What is a BLAST classifier? So a BLAST classifier uses the tool that's commonly used in bioinformatics, the basic local alignment search tool, which looks for similar sequences. It uses an edit distance-like algorithm to find sequences high similarity. Here, I take BLAST and any of the outputs that correspond to the highest scoring protein, I use as the output. Is it a phage virion protein? Is it not? Is it toxic? Is it not? And it performs the best. So the BLAST classifier performs better than all these machine learning models, which is a quite surprising finding, given that none of the other papers actually test against BLAST. So this paper actually adds that feature. Secondly, a thing to consider is the inference time. The time it takes for these models to run takes a lot longer for BLAST. So this is the benefit of using a machine learning model. If you're going to run a bunch of proteins to this, BLAST is going to be 20 times as long to actually run. The second question that I was able to answer using this tool is a protein a type of, or is a given protein, or what type of phage virion protein is a given protein? So this is a multi-class model. Again, using the BLAST classifier performs the best. I implemented other tools that perform this multi-class classification approach. So a feed-forward neural network, long short-term memory network, and a convolutional neural network. These are just different approaches using deep learning to perform machine learning, but from the high level here, they're different models that can be easily used to perform feature annotations on these proteins and easily implemented in this pipeline. Once a model is chosen, say you find a great model from the original training pipeline that you'd like to use now that it works well. That's what the prediction pipeline is for. Again, this is the novelty in this work compared to any prior work is that it allows for, it allows for metagenomic input, assembles that input, and output of that is contigs, or continuous, stitched together continuous fragments of contigs. Instead of using contigs or metagenomic data, you can also input bacteriophage genomes directly to annotate the open reading frames. That is where the OR finding comes in. So again, I use Phannotator here, a tool designed for bacteriophage genomes to find the open reading frames, and then I translate those open reading frames into the cognate proteins and use the trained models. One of the benefits of doing this in this work is I also introduce a graphical user interface that allows for scrolling along the genome and looking at any annotations from these models. You can use multiple models. Is it toxic? Is it a phage virion protein? Scrolling along those genomes and finding the different areas of those proteins. In the bottom panel, you can also specify the region that you'd like to look in and pull out the proteins that correspond to that area. So then you can blast it or do whatever else you'd like to do, maybe some secondary analysis to make sure that the models are working as desired. Last, just as a proof of principle here, I used an infrastructure for phage reference database to pull out 25,000 genomes and uncharacterized genomes. And from those, 6 of the uncharacterized genomes I used throughout the pipeline, just to show that the PhageScanner tool can be used to find phage virion proteins in these, again, or any other type. I also formed phage-encoded endotoxins that I won't be talking about today. So for all these uncharacterized genomes, there's differing numbers of phage virion proteins predicted. So this is just a high-level result from being able to find proteins in these genomes. Again, the counts are pretty interesting, but what is most interesting to me is instead where the proteins show up, the distribution of these proteins along the genome. So you can see the major capsid really collecting at the start of the genome, and some of the other phage virion proteins in the other areas. In general, this just shows that it can be used on uncharacterized genomes, and that subsequent analysis can be used to find further insights into these genomes. A future part of this work could be using this on characterized genomes, and then using that to just verify the findings from the genomic information. Overall, this is a machine learning pipeline that can be used to quickly test classification models for different annotations. The graphical user interface is presented for visual inference. And lastly, this is the first tool allowing for genomic and metagenomic data as input for PVP prediction. I want to summarize the dissertation and just bring it back to the high-level ideas here. The first question that I had is, can a digital microfluidics device be used to miniaturize and automate bacteriophage research? For that, I showed the PhageBox, which is a digital microfluidics extension that can be used to automate bacteriophage research and miniaturize it as well. The second question that I had is, can a space-efficient data structure be built and used to filter out sequencing reads from bacteriophages? Here I present, excuse me, in this water. I present PhageFilter, which is a software tool built using sequence bloom trees that can filter out reads corresponding to bacteriophages. And lastly, I asked the question, can machine learning models be used to annotate open reading frames in bacteriophage genomes? And with that, I presented PhageScanner, which allows for a graphical user interface to easily view any output from easily trained machine learning models. And again, this differs because the other tools are only using proteins as input, so it allows for a graphical user interface and extra ways to validate the process. I'd like to give thanks to a lot of people in this room. So first, thank you everyone for showing up here in person and anybody who's online. Starting off with my advisor, thank you, Marella, for advising me over the last few years, and all of the lab mates in the Living Matter Lab. I appreciate you. I appreciate the committee too for being here and for my comprehensive exam. So Robin Dell, Brian Layer, Chris Myers, and Bill Wagner, thank you. Co-authors that I've had the pleasure of working with, thank you for all the great insights and feedback. Also the 7 CU undergraduate mentees that I've had the pleasure of working with while here at CU, thank you. And lastly, I'd like to acknowledge the funding that I've had, so NSF, GRP, and Beverwood Spears. And with that, I will take any questions.
So we'll start with a few questions from the audience, which is not committee members. Yes.
Yeah, so when you were talking about the phage filter section, and you were discussing like the standard you were comparing it to for separating out genomes, one that came up a lot was FACS. And yeah. I'm not sure if that, like, are you talking about like actually flow cytometry or specifically virometry or?
That's a good question. And I'm hoping that people online can hear. So is facts that I'm referring to flow cytometry. So not flow cytometry. That's a good question, though. I should have maybe mentioned that. It's not the same thing. So this is a tool that was released in 2010. That was the first illustration of using Bloom filters to just filter out reads. It stands for, I believe, fast, accurate classification of sequences. So it's a little different there.
Thank you. I would probably go with that. Okay, so I thought it was really neat how your PhageBox expanded and filled a bunch of the gaps with the, was it Dropbox, the original?
Yeah, Dropbot, yeah.
Yeah, so I thought it was really cool how it picked up where that thing left off, but I'm wondering how the two things are integrated and how like you physically move a sample from one to the other. Do you have to like manually pipette it, or is there an automated way?
So I guess first the again, I'm not sure how many people are online, so if everybody could hear that, I won't repeat it, but
14.
Oh,
14, okay, I'll go ahead and repeat it. So with the PhageBox, how is this connected to the Dropbot and how are liquids handled between the devices? So this was actually a huge part of developing the device. It's like trying to figure out how we can add heating and magnetic modules to that original device. It turns out they have this adapter piece that on their GitHub that can connect that to any other potential external adapter. So that adapter piece essentially takes the pogo pins on the original device and extends them, so that way you can essentially add it to any other type of device created. So yeah, using that is essentially how I allow for the Dropbot to control the electrode, so any microfluidic operations, and then on the PhageBox controls the heating and magnetism. But yeah.
Yeah, I just was curious about bacteriophage therapy in general. I guess kind of two parts. Is it FDA approved or just in the research phase? And is there still the same possibility of bacterial resistance, or and if there is that just like another tool in the toolbox besides antibiotics? Or
Yeah, that's an incredible question. So the question is, are these phage therapies FDA approved, and is there potential for bacterial resistance in the phage therapies? If I understood correctly. I don't know the answer to the first one. That is a great question. So I'll look into it after this. For the potential for bacteriophages or bacteria to develop resistance to these therapies, that's an excellent question. This is something that I actually was reading papers about last week because I was thinking about it. I was like, wait, what happens if you get a phage therapy and then the bacteria comes back and you get another phage therapy? Well, eventually those are going to develop resistance. So there have been many papers showing that bacteria actually do develop resistances against these phages. But I did see one paper that showed that it doesn't develop resistance as fast as antibiotics, but overall, yes, they do develop resistance against them. And my guess is, this is postulating here, is the way that this can work at some molecular levels through CRISPR proteins, CRISPR-Cas9. So if those bacteria eventually develop a signature right inside of the CRISPR array for that bacteriophage, then any new infecting bacteriophage isn't going to work as effectively. That last part's a hypothesis, but yes, they do develop resistance.
Okay, thanks. All right, I have a question about your Bloom filter implementation.
Yeah.
So how long or short of a string or read do you need in order to accurately classify if it's from the bacteria or the phage?
How long, or sorry, do you mind?
Yeah, how long, like, does the read length affect how accurately you can classify it?
This is so like using like some type of like nanopore sequencing, for example, if it's long read sequencing or short read sequencing.
Yeah.
This is a great question. So how does long or short read sequencing, or long or short sequencing, or sequencing size and the read size in general impact the accuracy of the tool? This is something that I didn't actually add to the paper, but I explored. I got really curious because the only portable sequencers right now are nanopore. So why am I testing on short read sequencing data? I guess the goal would be to eventually be able to use it on such data, but it does work better actually on long reads. So the longer the read, the more characteristic that Bloom filter. So it seems to work better. I don't have any graphs inside the appendix that I do have set here, so that was that's a good question. But I'd be happy to share some of that information. It works really good for that.
Okay.
Oh yeah.
Yeah, kind of as a follow-up, even if it's more characteristics, that also result in a longer computational time, or?
It does. There's some information about the bloom filters that did not add to this presentation, given time and complexity. But the way that we actually create these bloom filters isn't by taking the entire sequence and putting that in a bloom filter. Instead, what I do is I scan k-mers along a read or a genome, and I add those k-mers to the bloom filter. If the read is longer, then it's going to take longer to scan those k-mers and add the bloom filter. So yes, overall, it will take longer time per read.
Oh, his follow-up.
It's going to keep bouncing back and forth.
What size camera do you use?
That's a great question. In fact, it's such a good question that I had backup slides just so there are two parameters actually. Let me actually show a different slide here. One of the problems is DNA sequencing machines are imperfect. So you might have some errors when sequencing reads. And the way that we get around this is by using those k-mers. So we can only use, we can require only a subset of k-mers to match. So given this read here, right, this like simulated read, essentially it should match up to that same read right there. It should be in that genome, but perhaps there's a problem with the sequencing machine. So if I expand that into all the potential k-mers here by taking that sliding window, then I can check if each of those are in the genome. So the first one is definitely not, the next two are. This allows for parameter theta. Okay, I want to introduce that slide before showing you the primary slide that I wanted to get to, because we have theta here. So theta is that adjustable parameter to allow for sensitivity versus precision, right? So to allow for that flexibility there. So the lower theta, the less k-mers must match. So here, k-mer size is uniquely connected to theta. So if I increase the k-mer size, this is going to decrease sensitivity and increase precision. And the same goes with theta. If I lower theta, this is going to decrease or increase sensitivity and decrease precision. So there's this flexibility with both of them. And doing this parameter scan, I use a k-mer size of 15 and a theta of 0.2. Yeah, thank you for the question. It's perfect. Was not planned.
Let's take two more questions,
Sorry, looking at that table, when theta was 0, you always had an F1 of 0.67. What exactly is F1? Is that tied to your sensitivity and specificity? Like,
yeah, that's an excellent question. So here, the 0.67 means you're essentially saying it's all bacteriophage, all those bacteriophage. So you imagine this should be a very bad F1 score. The problem is, in this particular parameter sweep, 67% would have resulted in true positives. So that is the baseline for this model. If you're characterizing everything. And again, F1 score here is a combination of recall and precision. So you can go lower than that score as seen on the right-hand side, but if you classify everything as bacteriophage.
And the last great question.
For the phage filter, do you have any comparisons with the other method as far as efficiency, or any more information regarding your implementation?
Yeah, there's a lot of information regarding the implementation. So there's a way to build the tree. I split the build and the query process of the tree. This was actually a huge part of the project that I didn't get into, but the original paper in the sequence bloom tree, one of their implementation is in C++. We moved that to Rust because I have a bias now. So I thought I'd throw that out there, especially on camera. The second thing is the way that the trees work are fundamentally different. So I have certain annotations in the nodes that are different. Now, when comparing to other tools for efficiency, those other tools I did in, let's see. I did show memory efficiency between the tools. So here, these graphs show the memory efficiency.
I'm in computational.
Oh, like speed. Yeah, I do have a speed test. That should be in the slides too. The speed test actually changes based on the size of these bloom filters, which is a whole discussion in itself as well. But it performs pretty well compared to some of the other tools. I do have. So here's just for classification tools, but in the paper, I also have a comparison of time between FACS and BioBloom tools, but PhageFilter performs pretty fast. Here I'm using a very large bloom filter. The larger the bloom filter, the longer it's going to take. I'm also decreasing the LRU cache size. So I'm saving the disk, I'm uploading the disk, saving the disk. That's going to take time. So even with me doing that and making sure it's super memory efficient, it's very fast. By balancing out the memory optimizations, actually balances out time too. So if I want less memory optimization, say I'm running on a laptop or a supercomputer, it's very fast. I think for us for that one.
Okay, so we'll go now into about 20-25 minutes from the committee with open questions with the audience. So try to have constructive questions at this point, and then we're going to have a closed door discussion with Dreycey after. So Robin, you're usually our guest, our first one to start with the questions.
Sure. So let's start with the phage filter.
Yep.
So you have to know ahead of time which phage
Yes.
you're looking for.
Yeah.
What's the extent to which phage are well sequenced, which that is a limitation to which these samples might have unknown phage that you couldn't possibly recognize?
Yeah, this is a great question. And it's one of the limitations I was thinking about with this type of data structure is if you have to know the bacteriophage genome beforehand, there's something widely different, like a twilight phage per se. How do you find that? So in here, I'm guessing that this wouldn't work very well for being able to find those unidentified phages. But another part of the slides that I didn't show is the implementation of the data structure. So not only do I allow for filtering for certain genomes, I think PowerPoint may have just crashed a little bit. I'm going too fast. Let's see, it might not actually have that. I guess I'll describe it though. So instead of just filtering for specific phages that are in the genomic sequence tree, what I can do is I can do negative filtering. So what I can say is, give me only reads that aren't contained in this tree, thereby only pulling out things that haven't been characterized or don't correspond to anything known. And the idea with adding this extra filter is maybe there's bacteriophage reads that we don't know. So what I can do is I can add a bunch of bacteria and genomic sequences from humans to this bloom filter, and then filter out anything that doesn't correspond to anything there. So hopefully only pulling out bacteriophages or the reads. I did not perform that test, but the tool does allow for the negative filtering. So if I had to, that would be the way that I use it, essentially pulling out anything that doesn't correspond to known genomes.
How similar are the proteins across the phages? Could you sort of combine some of the things that you did with the Phage Toolbox in terms of looking for ORFs and putting those into amino acid space where your alphabet is bigger?
Definitely.
And then using that in your bloom filter. I guess your bloom filter is really built for nucleotides, but could you imagine an amino acid version?
I've tested proteins, so proteins do work. So I think that's a great idea. Yeah, I think that actually would work. I had not tested that. That's an incredible idea. I haven't thought of that. But maybe just kind of building off that idea, maybe those phage proteins could be clustered, so that way we have a low sequence identity. Add a bunch of those to the tree, and then anything that's found from the ORFs could be classified as potential phage or something.
You might be able to reach a little further out evolution than things you haven't seen before that way.
Right, yeah. That's a great idea. Yeah. Okay.
So speaking of the toolbox, you had put up the different proteins and had to say in the ones you assayed and seeing how many times you saw those. And there were some zeros in there.
Yeah.
So
This one.
yes, do those particular guys lack a major caspid, or is that a failure of the data?
Failure of the models, unfortunately. What's kind of odd though is even for using like a blast classifier, I wasn't able to capture some of these major capsids. So I'm guessing it is the data underlying the models that just didn't have very similar proteins there. So future work could definitely be in trying to have these models generalize better. I think which is a hard task to begin with, but being able to still be able to find capsids that aren't similar to anything in the data set. All the phages should have capsid proteins though, for sure. Yeah. Yeah. Yeah, a major difference here too is this is the LSTM model that is using the black classifier. So here we see that there are major captains here,
Yep.
but not in the table. Yeah.
And then last question, and I'll pass it to somebody else. On your PhageBox, what can you imagine a project that is sort of ready to go on? What would you want to use it to if you had to jump in with both feet on something?
Yeah, I think I don't know, it's kind of exciting. I think the engineering part was pretty exhausting, so I was ready to work on a computer for a little bit. Where I imagine that going next is one, validating and performing PCR on the device. So currently what we have is just the hardware implementation. So showing a framework for doing PCR. So I think the very next step would be someone validating that PCR can work on the device, testing it out, testing out the framework, and where can it be used?
Yep.
That's a good question. I think what would be really interesting is trying to literally discover new phages using the device. So perhaps taking a soil sample and filtering out or extracting any microorganisms inside the soil sample, so filling it with some type of solution, filtering that, and then putting that on the device and trying from that point on to try and extract DNA from it. So you can imagine taking the liquid from the soil sample, heating it on the device to try and break up anything that's in there, and then perhaps mixing that with some magnetic beads to extract DNA, and then seeing if there's any nucleic acids that are extracted from that process. That's a very off-the-cuff idea that I come up with, so there's some gaps in that thought process, but yeah.
I'm good.
Well, just a quick follow-up on that. Do you think it would let you automate any tasks that are currently tend to be manual and a lot slower because you have those components there?
Yeah, that's a great question. What's the utility on the device as it currently
Or especially with being able to automate tests, that seems like a nice direction. But I was curious if you have any in mind.
Yeah, there's several. I guess this actually leads well with Robin's question. One is, I think, kind of funny and silly one that could probably be done without the device is just growing Mycobacterium spagmatis. So that takes several days, like 7 days. There's E. coli. I get a good mixture in 24 hours. So that was kind of a pain-inducing process trying to wait for that to grow. So I imagine what could be done there is you could grow Mycobacterium spagmatis by keeping both Peltier elements at 37 degrees Celsius. And then as it's growing, you could have, I also have an extension that goes on top of the top layer that I didn't discuss, but that could be able to look at perhaps the bacterial concentration. I don't know exactly how that would play out, maybe looking at the turbidity of the solution, like can we see any turbidity increasing to get a rough measure of the growth? I think there might be several other areas too that would work.
We also looked into bread or something, so genetic.
Yeah. So bread allows for modifying essentially the genomes of bacteriophages. So I think that could be done. There would be some modifications that are needed. So adding essentially electroporator to the device, which kind of executes bacteria and allows for DNA inside, which is needed for that protocol. And then I think one area that I'm particularly interested in is being able to create or synthesize genes. So there's an assembly method, Gibson assembly, that allows for creating new types of nucleic acids or being able to do gene synthesis. So you can imagine you have you can order small little nucleotides online. But what if you wanted to create a protein from scratch that nobody's ever seen before? You can order all of the underlying sequences needed for that protein and then perform an assembly of that on the device, very similar to PCR. And it's still required, the temperature cycles are still required, but I think that'd be kind of cool way to mix PhageScanner in with this is by finding some type of novel gene and trying to sequence it or synthesize it on the device. So that's a intrinsic one that I find kind of fun. But I think there are a plethora of different areas that it could be used for.
Sure. So PCR is pretty slow, right? On yours right now. I mean, in particular, the cooling times are super long.
Yeah.
I wonder if you could use your little reservoir, instead of dealing with evaporation, to use it to like cool the solution off by adding like cold water. You think that would work?
For sure, yeah, that's a really good idea. I think the only potential danger is if it cools it too far.
Yeah.
Or if it changes any of the reaction kinetics.
You didn't ever talk about host response to filling somebody with a bunch of new viruses.
Right.
Like how do you control for that?
Yeah, that's a really good question. So I don't know. I don't know. Like if you were to give a phage therapy and there's a bunch of virus all at once, I don't know. But again, off the cuff, just hypothesizing here, I think that there would be an innate or an immune response to that immediately. So maybe eventually, like I think there's probably a dose that's too high. Whereas if you give that dose, your body immediately responds to it, kind of giving an adverse reaction to what the therapy is supposed to do. So killing off the therapy when it should be killing off the bacteria.
Like I'm cute for that woman. I mean, she had systemic infections, so they must have just pumped her full to get to every single little piece of her body. I mean, did they?
They open up usually when they.
Oh, it was direct inject.
Usually they do if the patient is so critical.
Okay.
They apply doctor directly.
Yeah.
Wound locally. They do surgery if they have to.
Yeah, I think it does still make me wonder about the efficacy of these cocktails later on. Because even if you inject at the side of the wound, eventually your body's going to respond to leftover bacteria. Yeah.
One comment, so long read is bad for metagenomic sequencing because the longer the read, the fewer bugs you sample in your sample. So short read will sample more things, just less of each, where long reads you'll get very long. And since your sample is almost going to be, what was the percent of phage in your test samples?
In the test samples, I varied it. So I would have anywhere from 5 to 5% to 0.1.
Okay.
Yeah.
So yeah, so you would be sequencing almost getting very long reads of human or whatever. So long reads don't work great for metagenomic sequencing. Can you go to slide 31, please? So in particular, this size versus number of genomes, I'm a little concerned with that experiment because, as you know, Bloom filter is a fixed size that is a formula of the number of this right one where you have your Bloom filter is fixed size. And so I'm curious, so a Bloom filter size is a combination of the number of insertions and the expected error rate.
Right. Yeah.
Are you keeping the error rate constant as you're adding numbers? So adding number of genomes, you could just be increasing your error rate. So you would need to then compensate that with increasing number of hashes or increasing the size of your genome. So is error rate constant on this graph or is it? Because
this is a very detailed. It's like if I add more genomes, the error rate of these Bloom filters is expected to increase.
Right.
So this does keep a constant error rate and a constant Bloom filter size. So that's a good point.
So you can't do both. You can't keep constant Bloom filter size and constant error rate.
The way that I do this is because the genomes are at the leaf nodes of the tree, there's not multiple genomes per Bloom filter. Each genome is given its own individual Bloom filter and results at the leaf nodes of the tree. But the root node, I think, is the big point. The root node does have an increase in error rate, which I think is a really good point. If I want to increase the Bloom filter size to reduce the root node error rate, then that would increase the memory consumption. Hopefully not above 2 gigabytes. So this is a very low, this is like the minimal expectation for how low can we get the memory? How low can we go? So here, the idea is this would be very low, but I think that we could still expand the size of this and still stay below the 2 gigabyte requirement for the Raspberry Pi at least.
And what drives the structure of the tree?
Yeah, this is part of the slides that I took out just for time. So how do I build a tree? I think is
so Kraken does really specific tree of life kind of things, and then how did you when you bifurcate, what is right, what is left?
Yeah, so this is a really good question. I'm going to skip to the slides. I think using taxonomic information would be incredible for this tree. In building it, I don't do that. And no other authors do, so potentially that future work, that's something could look into. What I do is I follow a very similar process to the original authors. So they, again, use...
The original authors being Kingford?
Keyford, yeah. And they used RNA-seq data here. So they do is for each of these data sets, again, they build a Bloom filter. So when they add another data set, there has to be an internal node added. That internal node contains the union, so it's added like that. Okay. So then how do we add a newer node? This is the real kicker of how this tree is built, because there's some complexity here. And I think further inference into the balance of the tree is needed. This is actually a self-criticism from this. This is something that I've discussed with Marola and others. I think is still needed for this work. So when you add a new Bloom filter, after you have this basic tree, how do you decide where this new data set will go? Well, because you can't add it to the root node, this is an internal node, you have to choose on whether to go right or left. And that's based on the Hamming distance. So the lower Hamming distance is used to choose the direction to go and add this new Bloom filter. So
Hamming distance of the bit vectors?
bit vectors, yeah. And yeah, this is the Hamming distance of 2. I didn't add that here, but I remember that particular slide. So then this would be added to the right-hand side, because those have more similar Hamming distances. So that is how the tree is built.
And then what is distinct with your implementation versus Fax?
So Fax uses a single Bloom filter, and I think it adds all.
I'm sorry, I didn't mean Fax. I thought that was Kingsford's original Bloom filter tree.
Oh yeah. So like this one, there's several different implementation level details that are different. One that I believe is the novelty inside the tree is using it for a completely different area. So here, testing whether or not a transcript is inside the tree, and then for us, testing whether or not a read stems from a genome. So when it comes to the implementation level, I had to make sure that all the nodes in the tree keep count of the reads that map there. So that allows me to say how many reads map to each genome. And then also I added another data structure that kind of wraps around this that I don't talk about in a lot of detail, but the LRU cache and the read buffer, both of those are very different than this. So they don't use any memory optimizations in this tree.
Can you go to slide 29?
Yeah.
So my question is going to be, how do you have a good F1 score and low classification?
I think.
So you have very good F1, but very poor classification. How are those? Didn't you say, wait, so it is, the bottom slide is F1 classification, and the top is F1 of what? Of individual reads?
Yeah, filtering individual reads.
So how do you have good filtering but poor classification?
I think there's a huge way that this can be optimized. Currently, what I do is for all those reads, if any of those, so for multiple reads, when I filter those out, if any of the genomes contain that read, I consider that a true positive. For classification, that read might come from many genomes, and I don't choose which genome is best fit for that read. So I take the list of all genomes that read maps to, and I say those are true genomes. So a way to really improve the score would be to take only the top scoring genomes. So that's part of the reason why it doesn't perform that.
Or like just find the unique sequences in each and only use those for classification.
Yeah.
Sorry, I'll stop.
I'm gonna press the link to Chris and then we can do.
Yeah, I can be really quick. Dreycey, great presentation. It's very clear. I think most of the questions at this point have been asked and answered. So most of the things I have remaining are more appropriate for the closed session. One question I will ask though is why the embedded device? Why my Raspberry Pi? What is the motivation behind putting it on that device?
Yeah, one of the motivations was I realized with this type of sequence Bloom tree, I could be able to only populate certain nodes in memory at once or use that LRU cache. And in addition to that, I was thinking a lot of these bioinformatics tools don't really have a huge emphasis on being able to run on embedded devices. I have seen like certain assembly tools that can be used on embedded devices and low memory.
But did you have an application line where an embedded device would be essential? And why an embedded device do you want to do in the field? I mean, what is it?
Yeah, field operations, and in particular nanopore. So like the current sequences that are portable right now would be nanopore. But again, if that doesn't work the best for sequencing metagenomic applications, then maybe the future of these devices, I fully expect these to become portable. So if we use some type of field application such as sequencing soil samples for bacteriophages, then we can immediately filter those out and discard any reads that don't correspond to.
Is there any performance improvement from this and from putting it in a specialized hardware? Are you seeing any performance improvement, or is it just about the size and portability?
Performance, by putting
By
on
putting on
hardware.
hardware versus, general purpose.
No, the tool performs very well on like a laptop computer. So when using this, like the hardware of a laptop, it performs very well compared to other tools in time and memory. But the small embedded devices are so slow that there's a huge memory disadvantage.
Disadvantage, yeah. So it's not the usual put it in the hardware to make it faster.
No,
Like
it's-
you were trying to, I guess if you map the hardware like an FPGA or something, you might see more sustained improvement, but you're really just running the code on a different device.
Right.
And it has memory limitations, which is what drove your decisions.
Yeah. I think one additional thing is, right now I compile this on the Raspberry Pi, but I think an interesting future piece of this work could be to use it through something like Serial, so that way a portable device could send sequences through and then just get back sequences that correspond to bacteriophages. So maybe that would be a better usage of this, because then any type of sequencer could use it.
Okay. I think I'm ready to move close. If you want to let the audience talk a little bit.
Yeah, so thank you everyone for being here. I'm gonna kindly ask you to leave the room, take the food with you if you need to leave, and we're gonna continue the closed session and let you know when the liberation comes. Are you recording?
It's
Oh.
still recording. Should I stop?
Yeah,
Yes.
Okay.
Yes.
and then log off.
You should probably